This video is a video of the protein synthesis lab. So we're going to start off with the first step of protein synthesis, which is transcription. And transcription takes place in the nucleus. So we're going to pretend that this manila envelope is the nucleus, and we're going to go into the nucleus. And in this nucleus, we have a DNA sequence. Now, we're going to pretend that this DNA molecule is already opened, and it would be opened by an enzyme called DNA helicase. This is the same enzyme that opens the replication fork in DNA replication. But this is going to be different. This is going to be transcription. And instead of DNA polymerase building the new strand, now we have a different enzyme, and it is called RNA polymerase. So we're going to pretend that we are RNA polymerase, and we're going to make mRNA. So this is going to be our messenger RNA, and we're going to come up with the complementary sequence. T goes with A. A would go with T if this was DNA, but there is no thymine in RNA, so it's going to go AU, and then C is complementary to G. G is complementary to C. G would be complementary to C. T is complementary to A. C is complementary to G. T is complementary to A. And there's two A's. C is complementary to G. T is complementary to A. C is complementary to G. T is complementary to A. A is complementary to U. And there's two of them. And then T, go A, C. And this would be U, A. So this mRNA is complementary to our DNA strand. Everywhere where there would have been a thymine is now a uracil, because RNA has uracil and it does not have thymine. We have just simulated transcription. The enzyme that builds the mRNA is called RNA polymerase, because it's building a polymer of RNA. Transcription takes place in the nucleus. Now, we're going to leave the nucleus now. DNA never leaves the nucleus, and that's so that you'll always have the recipe you need for making the protein, and it will be protected from all of the enzymes in the cytoplasm, and it will stay where it needs to be. Kind of like reference material you're not allowed to check out of the library, so it's always there for you. So this is an mRNA, and it is the temporary recipe. It's kind of like a copy. Now, that one segment of DNA we would have called a gene. If we look back at this would be a gene. It codes for one protein. Of course, real genes are much longer than this, but we're going to pretend. And so this mRNA has a really important job. First of all, it can leave the nucleus, so DNA can stay there. So it brings the message out of the nucleus to the ribosome. These groups of three bases are called codons. In DNA, the three groups of bases are called triplets. These are codons. They are on mRNA. So now I'm going to find my ribosome. This yellow thing is the ribosome. And there is an A site and a P site. In the A site, the new tRNA molecule matches up and brings an amino acid. And the P site will be holding the growing polypeptide chain. So now I just want to show you, I have a whole bunch of tRNA molecules that I have already charged in advance. So on one end of the tRNA molecule is a group of three bases that will be complementary to our codon. This group of three bases on tRNA is called an anticodon. On the other end, we'll always be carrying a specific amino acid. So every time there is a CUC on the tRNA, there will be glutamic acid carried there. So this is a charged tRNA molecule. Without the amino acid, it's considered uncharged. It can it can go back and grab more amino acids later after it drops its correct amino acids off. So it goes back and forth between being charged and uncharged. So the first thing we need to do is look at our first codon, and it is AUG. We are going to translate this mRNA sequence. So now we are in the cytoplasm at a ribosome, and we are going to be performing translation. So we need to find the anticodon that is complementary to this codon on mRNA. So we are looking for UAC. So UAC is a tRNA molecule that's carrying methionine. So A and U pair up, U and A pair up, G and C pair up. Then we slide this over, and then the next tRNA comes in. So CCA, we would be looking for GGU. So this is going to be our next tRNA molecule. And we are going to lift up the first amino acid and bond it to the second amino acid. Now, truth be told, there is an enzyme in the ribosome that catalyzes the reaction to make this imaginary peptide bond, but we kind of can't simulate that. So this tRNA is uncharged and it leaves, and it would go pick up another methionine for later. Now, this slides over to the P site, and now we need to find our next tRNA molecule. So GAA, we need CUU. So here comes our next tRNA. And we lift up these two amino acids and attach them to the new amino acid, forming our peptide bond. And of course, our imaginary enzyme in our ribosome helps catalyze that reaction to get them to stick together. Otherwise, they'd come in the right order, but then they'd just fly away. So now we are going to be finding our next tRNA molecule. So GAG would be CUC. So we come in, match our next anticodon to our codon on mRNA, and we're going to lift up the growing polypeptide chain, attach it to the new amino acid. This tRNA leaves, and then the mRNA slides over to the P site. So now we have AUU, so we need UAA. So that's going to be Mr. isoleucine, which actually has the three-letter symbol ILE. We're going to lift our growing polypeptide chain up, and we are going to attach it to our new amino acid. Our tRNA molecule leaves. This slides over, and we have AAC, so it's going to be UUG. If you look, this anticodon, which is on our tRNA, is complementary to our codon. And the new growing polypeptide, whoa, it broke apart. Our new growing polypeptide chain is going to attach. And our tRNA is going to move. And this last codon is actually a stop codon. So there'd be an enzyme that says, hey, stop here, and we know that it's the time to stop, so then we would just remove this polypeptide. Now, granted, in real life, the polypeptide would be longer, but I just wanted to put that here for you so that you could see the order. This is the first one. The first one is at the top. The last one is at the bottom, because one of the questions on your lab is going to ask you to write the polypeptide sequence. So a polypeptide is many amino acids stuck together. In a real protein, it would be a lot longer. The shortest one that I can think of is 52 amino acids. But when you write this, if we were doing this in school, you would just tape this down and you wouldn't have to write it. But on the computer, you're going to have to type it in. So you're going to type the first three letters. You're going to type MET, and then put a dash with no space before or after the dash, and PRO dash, no space between the next one, GLU dash GLU ILE and ASP. Now, when you do this, you should not put any spaces before or after the dashes, and you should use all lowercase letters, and you should not put any spaces before or after the last amino acid in this chain. Just understand that this is just a short simulation. So DNA to mRNA was transcription, and it took place in the nucleus. mRNA to amino acid sequence is translation, and of course the polypeptide would fold into its three-dimensional shape based on the parts of the protein that are attracted to water would be folded onto the outside, and the parts that are repelled by water would be in the middle and away from the water. So just a quick reminder, this is our mRNA. Its important job was to bring the message out of the nucleus, and it is able to leave the nucleus so DNA can stay there, and it has anticodons that tell the tRNA molecules which order to come in and which order to bring the amino acids. The tRNA's job is twofold. One, it matches its anticodon to the codon, and that ensures that the amino acids get in the right order, and on the other end, on the top end, it carries an amino acid, and a very specific and correct amino acid, to make sure that they end up in the same order. And there is an enzyme in our ribosome that helps bind the amino acids to each other so they don't end up all in the right order and then just floating away, so that the peptide bonds form. Now, one of the questions is going to ask you to compare the first DNA triplet to the first tRNA that came in. So if we look at this, TAC was complementary to AUG, and I want to find the tRNA molecule that was also complementary to AUG. So it was UAC, right? These three bases, this is the tRNA, this is the anticodon that was complementary to this codon. So they're asking you to compare the first DNA triplet with the three bases, the first anticodon. So if each of them are complementary to the same sequence, this is what they would look like if you put them side by side. So I don't want to give you the answer that I'm looking for there, but I want you to look at the sequence and notice, how is the sequence of the first DNA triplet alike the first anticodon, and how is it different from the first anticodon? Because that is one of the early questions in the lab. This has been your protein synthesis lab simulation.